The overall goal of this procedure is to measure associative memory using the adult Drosophila olfactory shock assay. This is accomplished by first collecting flies under carbon dioxide anesthesia. The second step is to expose the flies simultaneously to an electric shock and a first odor, A, in the T maze. Next, the flies are exposed to the second odor, B, without a shock. Finally, the flies are taken to the choice point in the T maze, where they are exposed to both odor A and B simultaneously. After 2 minutes of distribution time, the flies that chose odor A and those that chose odor B are collected separately and counted under carbon dioxide. This allows calculation of the performance index for each odor. The final PI is obtained by averaging the PI for 2 separate experiments, one in which odor A is shock paired and the second in which odor B is shock paired.
This method can provide insight into the effect of the internal state of the animal on memory, for instance by varying its age, diet and sleep. It can also be applied to other systems. For instance, it is analogous to rodent olfactory-mediated fear conditioning assays, with the genes and mechanisms of learning being highly conserved between insects and mammals.
Visual demonstration of this method is critical as preparing the flies for the assay and then moving the flies into, within and out of the T maze is difficult to learn and not easy to appreciate how to do from just a written protocol.
To begin, grow flies on a standard food diet under a 12-hour